Vedhaan channel.
Now we are seeing about the placental barrier, and that placental barriers, placenta is strongest barrier for maintain the fetus immunity response. A placenta is one of the barrier. It means it is important one which one it's occur between the fetus as well as to the mother. It will be used to prevent the infections. It will be prevent the disease also. Means it is improving the immunity by the placenta. So second thing is the placenta barrier consists of the tropoplastic epithelium covering the villi, and that second one is the chorionic connective tissues, and third one is the fetal capillary endotheliums. The three things will be act like a placental barrier. And it is another one is a selective barrier is another functions of the placenta. It is one of the selective barrier also. And what is a selective barrier means? It will be a placenta act as selective barrier from maternal blood, protein and microbes such as bacteria and virus. If mother's blood getting to that whatever the nutrients and blood sets coming from the mother's blood, that will be selected which one is suited for the fetus, that only will be selected by the placenta, then it will be sent into the fetus. So that is the selective barrier. And some of the virus can cross the placenta barrier, including rubella virus, zebra virus and cytomegalovirus. So here if we see there's some virus means it will be cross the placenta. What and all means it is a rubella virus and zebra virus and cytomegalovirus. Like that virus it will be cross the placenta and it will be affect the fetus also. So, in the placenta barrier function start from 13th week of gestation and increase the level of IgG antibodies in the third trimesters through the human placenta for providing protection of fetus in the uterus. Here in that placenta barrier functions, it is started at 13 weeks of gestation. At the same time, it is the IgG antibodies also will be having increased during time of the third trimester. The human placenta, it will provide in the IgG antibodies only it will be provide the protections of fetus in the uterus. So then this passive immunity and after delivery of the baby has to keep in that low level of placenta. It means that and the passive immunity occur after during time of occur after the delivery of the baby, while that mother the baby has to keep in that low level of the mother's abdomen. So that time the placenta blood, and that means at bottom of the blood placenta blood, it will be coming out of the coming out of the womb. At the same time it reaches to the baby through the umbilical cord. So it will be having that complete blood it's coming from the placenta, no? That blood it is giving to the immunity power of the baby after delivery of the fetus and after delivery of the baby, and it will be having that so the large amount of immunity response blood receive the baby from the placenta bed. The two pattern the fetus hand baby receive the passive immunity. The two pattern will be there. Two method will be receive the baby's immunity. Baby's immunity. One thing it is the blood. Another one is the breast milk for the several month, means that is having the 3 months. The nutrients, whatever nutrients and immunity power received from the mother's blood, that will be keeping the 3 months or 3 to 4 months of the baby. So after birth, providing of mother's immunity to see the infant life, it is IgM antibodies is largest size cannot cross the placenta. So in this IgG we saw previously that IgG it's occurred during time of the intrauterine life, and IgM it will be antibodies occur in that extrauterine life, means after the delivery of the baby IgM antibodies will be. This is a larger size, so it can't able to cross the placenta. The placenta has some method to transfer the materials between the maternal and fetal circulation. it's having the some methods through the placental membranes. The following methods are there. One is the diffusion, second one is the active transport, and third one is the placental infection, and fourth one is the endocytosis, and last one is the exocytosis. These are all the methods will be having during time of the transportation of the circulations maternal and to the fetal circulations. So now we'll see that first one is diffusion method. In the diffusion method, the substance are transferred through a membranes from higher concentration to lower concentrations. In that substance transferred through membrane. Here membrane will be there in between. Here in the higher concentration to lower concentrations, that substance are transfer. Examples if you are seeing is water, oxygen, carbon dioxide, urea, and certain simple amines, it will be transferred from higher concentrations to lower concentrations. Example if you are seeing means water, it's coming in that water or air will be there. So it will be transferred from higher concentration to lower concentration. So it will be balanced from in and out. That method it will be followed here. First method, and second method is active transport. In that active transport, the substance or transfer from low concentration of maternal blood into the high concentration of fetal blood. It occurs against pressure. Here you will be having that lower concentrations will be there in the one place, and at the opposite place it's having the higher concentrations. Previously we saw that in that diffusion method it is having higher concentration, lower concentrations. It will be having that normal pressure. But if you are seeing that active transport, it is having that against pressure. That is the low concentration to high concentration will be there. If you are seeing that example, transport of iron and ascorbic acid from the mother to the fetus. If a sufficient amount, it will be there in the fetus of iron and ascorbic acid, but in that insufficient will be there in the mother place, but it is absorbing that iron and ascorbic acid to the fetus. So this is the active transport of methods. And the next third one is placental infection. The placental infections, protozoal and bacterial infections are affected the placenta, but it did not affect the fetus. What is it? Protozoal and bacterial, some infections, it's affected the placenta, but it's not transferred to the fetus. And viral infections may pass through the placental membranes, and fetus gets affected without affect the placenta. If it is having some infections is not transferred, and some it will be transferred to the fetus, but in the time the placenta will not affect any infections, but in the time the fetus it's affected due to the virus and bacteria. And next thing is cell engulfs external materials by incoagulating its cell membrane to form a vesicles. This is will be the some cells are it will be engulf the external materials. That is it's coming to that endocytosis. A cell engulfs external materials by incoagulating its cell membrane to form a, it is where vesicle formations will be occur. That is the IgG, the immunoglobulin is taken up the endocytosis from maternal circulations. Here, if you are seeing that endocytosis is the process by which a cell takes substance into itself by engulfing them with its cell membranes, forming a vesicles. If you are seeing that endocytosis, this is a process. The molecules are entered inside of the cells and membranes, membranes, and it will be engulf the cells. This is the process of endocytosis. And if next if you are seeing that exocytosis, it will be having the Release of molecules within the vessels to the extracellular space. If you are seeing this image, you can able to understand that exocytosis. This is the first process. First image is the first process. The secretory vesicles to that will be so molecules or it will go to the cell membrane of the cells. Next, it will be produced. It's out of the cells. So this is the exocytosis, and this is exocytosis is the process by which a cell removes or secretes substance out of the cell by fusion of vesicles with the cell membrane. Here, engulfing the cells, it is the endocytosis. Exocytosis means it will be spell out the substance that is called that exocytosis. This is endocytosis, it will be engulfing, and this is exocytosis, it will be a secrete spell out the out of the cells. There is the exocytosis. And this next method is the examples of that exocytosis is IgG immunoglobulins are transferred to the fetus from the placenta by the exocytosis method. And next last one is the breaks between cells. The chorionic will is getting break and allow for the direct transfer at cells. Using the chronic release is getting the break. It will be getting the break. Automatically the chronic will be getting the break and allow for the direct transfer of the cells. This is the sensitization of an Rh negative humans from the receipt of erythrocytosis, erythrocytes from her Rh positive fetus. If in that cells, if a mother is having Rh negative cells, it will be getting that break down the cells, and next it will be passed, it will be enter into the inside of the fetus cells. It will be having that fetus, it's having the Rh positive. So here it will be if it is mixing the mother's and fetus blood, that it is having the condition all are knows that erythrocytosis fetalis. And this is the layers. If you are seeing that in that green and cytotropoplasm, in cytotropoplasm, and next blue one is the basement membrane, and that maroon color is the connective tissue, stroma, fillus. These are the four layers are will be there, and it acts like a placental barrier. And this is the importance of antibody during pregnancies, maternal and antibody, and breast milk antibodies. This we saw no IgG and IgA, and this is important methods. Thank you.